welcome to Israel again. It's very nice to see you here again with us. And I would like to talk to you about your subject of interest, which is the genetics of epilepsy. But can you start by telling us about yourself?
Well, thank you for asking me to participate in this. I was born in Melbourne and I was educated at the University of Melbourne, and I got interested in neurology in medical school. And I remember my first job as an intern was actually the intern on our neurology service at our hospital in Melbourne. And I could see that the head of the unit, whose name was Peter Bladen, was doing things that kind of others didn't seem to be doing or talking about in epilepsy. And Australian neurology was largely steeped in the British tradition, and at that time the UK neurologists weren't, for the most part, terribly interested in epilepsy. And Peter Bladen saw this as wrong because it's such an important and common affliction, particularly in children. And he started a surgery program in Australia and got me interested in epilepsy, got me fired up in it, and suggested I do my fellowship in Montreal at the great Montreal Neurological Institute with Fred Anderman, which of course was the way we connected the first time. And I had a 3-year fellowship in Montreal, which was just a wonderful experience. And I tell all my trainees the fellowship years are probably the most exciting, in the sense that you, at that time in your life, you know everything about everything, or you think you do, and you're generally relatively unencumbered by the other pressures of life, and it's a time to let your intellectual activity flow. And I was extremely lucky, being in the right environment with the right people. After that, I returned to Melbourne. I was never formally trained in genetics, but Fred Anderman's wife, Eva Anderman, is a neurogeneticist, and she got me very interested in the genetics of epilepsy. And I started doing some studies in Montreal during my fellowship, but I could see that it was a valuable area to get into. And I started that as my main research interest when I got back to Australia in the late 80s.
And you've been in that since then? That's
right. It's been my
But do you still practice neurology outside epilepsy?
Largely not. I tried to. Again, as I said, one thinks one knows everything about everything at certain periods of one's life. But it became clear to me that I was losing the feel and the touch of dealing with other common problems like stroke and Parkinson's disease and dementias, and I then more or less have restricted my practice to epilepsy.
And can you tell us about your connection to Israel? So you've been here for several times?
Yeah, I've been here several times. My major period here was a 3-month sabbatical in 1999, and I spent 3 months here at Ichilov. And the idea was then to see whether some of the novel findings that we'd observed in the largely Anglo-Saxon population in Australia, in terms of familial patterns of epilepsy and some very early gene discoveries, might be replicated in the Israeli population, which is ethnically, first of all, heterogeneous and very different to what there was in Australia. And that sabbatical was a spectacular success from the scientific point of view. We achieved all our aims, but also from the sort of sociological point of view. I had a wonderful time basically touring Israel and going into people's homes, and I really learned a lot about the socio-cultural aspects of Israel, and really the socio-cultural aspects of epilepsy as it's experienced within people's houses. You get a different view when you see it from inside the house compared to the consulting room. And since then I visited Israel periodically, generally about once a year, to keep the project going, and it's been very successful.
Yes, it was indeed. Several papers that were published based on the studies that were done here, and we are very grateful to you for doing this with us here. So epilepsy is an important part of the neurological sphere now. Can you define epilepsy for us?
Legally, in most countries, it's been defined as two or more unprovoked seizures. But in fact, in the last couple of years, there's been a tendency to make the definition a little bit more broad and to have it to be defined as a lasting predisposition to, or an ongoing predisposition to have seizures. And it's rather difficult to say where epilepsy begins and stops. Pragmatically, I don't see a great difference between a person who has a single seizure compared to somebody who's had two seizures at the beginning, but this remains an area of debate. But it's still useful to think about it as unprovoked seizures and either a second one or a predisposition to have more.
So these seizures should originate in the brain. So, for example, if a person has hypoglycemia several times with loss of consciousness and maybe seizures, you wouldn't call that epilepsy, or would you?
No, because the word is unprovoked, and here the hypoglycemia is a provoking factor. As we know, every human being can have a seizure given enough stimulation, and hypoglycemia is one very potent cause. And as we know, it was a now historic so-called treatment for severe mental diseases. We used to induce hypoglycemia using insulin to provoke seizures and have a sort of primitive form of ECT. So I think the unprovoked is important, but again there are shades of grey there in terms of when something is provoked or unprovoked.
And so it depends on the phenomenology, on the phenomenon of what one sees.
Yes.
So this begs a question. So if you see, and we saw this in different cases, for example in GLUT1 deficiency, where abnormal movements can occur, which are not reflected by EEG changes. So these are movement disorders. So these are seizures, but not epileptic seizures. And these same patients may have also epileptic seizures and may have epilepsy. So again here there is a distinction which is not clear to me.
Yes, it's a tough area in the so-called borderland of epilepsy, which was a very useful term that Gowers used over 100 years ago. So as you've indicated, and you know well, there was a literature really trying to firmly distinguish epileptic seizures, pragmatically defined as seizures associated with electroencephalographic changes, from non-epileptic seizures, of which there are many, and movement disorders are an important cause of that, paroxysmal kinesogenic choreoathetosis, etc. And those in the epilepsy camp and the movement disorder camp were quite keen on making these distinctions. But the recent genetic studies have really made liars out of us, in the sense that we can see the same phenomena within the same family and indeed in the same individual at different eras in their life. So, for example, in a disorder that's now known to be due to a gene called PRRT2, the individuals may develop unequivocal epilepsy, infantile convulsions, around the age of 6 months, and then typically around the age of 10 they may develop paroxysmal kinesogenic choreoathetosis, where there is no epileptic abnormality. It responds to carbamazepine, which is of course an anti-epileptic drug. So it becomes an interesting issue of definition as to what fundamentally is epilepsy, and I don't think we have a firm answer to that.
And even the GLUT1 deficiency, this is a case in which actually this is hypoglycemia as a cause.
Perhaps, yes. Certainly glucose delivery to the brain is restricted, and you get seizures in that, you get paroxysmal movement disorders, and you also can get fixed neurological deficits, including cognitive impairment, including cerebellar ataxia, including fixed movement disorders.
So you call it epilepsy, although these are really hypoglycemic in origin. And before you say that hypoglycemic seizures should not be included in epilepsy.
Well, I guess one can argue there that hypoglycemia induced by an insulin overdose or perhaps an insulinoma is a provoked type of event, whereas if you've got a genetic predisposition where glucose entry into the brain is restricted, you don't need an extra provoking factor. But I agree, it's a grey area.
So before we go in more depth into Mendelian epilepsy, what do you think is the contribution of genetic causes to epilepsy? Are most cases with the sporadic form, the non-familial form, also have a important genetic contribution?
So my belief in that is yes, but the data is still not finalised. So to put this into perspective, how do we come to that hypothesis? Well, it's well known that there is an increased frequency of relatives with epilepsy amongst probands or index cases with epilepsy, but that rate is not that high. The so-called recurrence risk ratio, in other words, the frequency with which epilepsy occurs in relatives compared to the general population, is about threefold. So the general neurologist will see many patients with epilepsy. Most won't have a family history, but some will, and I would expect that figure of 3 kind of fits with what one's general expectation is. But it's not the frequency of family history that one sees in classical Mendelian disorders. For example, Huntington's disease or in muscular dystrophies, etc., where the recurrence risk is much higher. So what's the data that then suggests that many cases of epilepsy have genetic component? And that comes from twin studies. And in twin studies, the concordance, that is the likelihood that one twin, the likelihood when one twin has epilepsy that the second twin will also have it, is remarkably high. And it's much higher than many other diseases that have a genetic component. And for example, in the generalised epilepsies, the figure is about 0.7 to 0.8, which is extremely high. So this speaks to the fact that most forms of epilepsy have so-called complex forms of inheritance where multiple genes contribute, and one doesn't often see first-degree relatives that have the disorder. And that in part is the solution to the paradox. But a second, quite unexpected discovery has been the frequency of so-called de novo mutagenesis. That is, in individuals where there is no family history whatsoever, they develop epilepsy, and we now know that in many of those disorders, and I'll talk about which in a moment, the disorder is in fact a de novo mutation.
So is it a somatic mutation, or is it inherited then?
It can be both. So the best recognised type is where the mutation occurs in either the sperm or the egg, and the individual is a heterozygote. 50% of their DNA copies have the mutation. And the best known example of that is the so-called epileptic encephalopathies, which are a group of very severe epilepsies in children, which can be due to a variety of extraneous causes like hypoxia, ischemic encephalopathy, and a variety of other insults. But many of these children had absolutely no cause identified and typically had a normal MRI. And these were a big mystery. And what's now clear is that the major identifiable causes in these is de novo mutagenesis. So one finds a mutation in the child and there is no mutation in the parents. And that's quite important for genetic counselling because by and large, with some subtle exceptions, the recurrence risk is low. The chance of having a second child is low. Now, such de novo mutations can occur as I've said, in the germ cells, either the sperm or the egg of the parent, but they also can occur in the early embryo. And in such cases, the expression of the disorder may be limited to one particular organ of the body. So, for example, obviously in neurology we're interested in brain disorders, but some hitherto unexplained disorders are now shown to be due to post-zygotic, after fertilisation mutations that essentially affect the brain. And the best known example is hemimegalencephaly, where a child is quite severely abnormal and has one hemisphere that is extremely severely deformed with cortical dysplasia, and they commonly have intractable epilepsy and intellectual disability. And by studying the DNA in the brain and in the blood, one can show changes in the brain that are absent in the blood. And that is so-called somatic mosaicism. Now that's tough to identify unless you have brain tissue. So far we largely only know about that in severe epilepsies that undergo surgery, but there's no reason to suggest that this may not be a more common mechanism. And of course it's the mechanism in cancer where you get new sporadic mutations. So when one puts together the emerging sort of new genetic mechanisms and the genetic epidemiology, particularly as shown by twin studies, there's a case to be made that much more of epilepsy is due to genetic causes than we hitherto realised.
So how is this reflected in GWAS, in the genome-wide association studies?
So GWAS is a important technology that emerged around 2007 to look at the so-called common variants. These are not necessarily novel mutations, but changes that we all carry in our genome, and look at which common mutations predispose to disease. And the GWAS studies in epilepsy have been relatively limited because so far we haven't had very, very large sample sizes. And the biggest study was in fact a study that we coordinated last year through the International League Against Epilepsy with nearly 10,000 cases. And this showed a small number of hits, but we now know from studying other diseases that we need to get even larger sample sizes to get more hits. The problem with the discoveries with GWAS is that the effect size, in other words, the amount that the variation predisposes to the disease, is actually very low, like 1.1, 1.2 or even 1.05. It just is a very minor risk allele, and it would seem that one would need very many of those to result in the phenotype, and we're not quite sure yet how important common variation is to epilepsy. It's clearly quite important in some other disorders, but whether it's the key mechanism in epilepsy remains to be determined.
So you mentioned before that in some of the sporadic cases there could be a genetic contribution by several genes. Is there any example of that? Because the alternative would be that it's not polygenic, but rather interaction of one mutation with environmental factors.
Yeah, that's a good question, and that's one of the sort of major challenges in genetics per se. And that hypothesis is not interrogated by GWAS, but is interrogated by deep sequencing, so-called whole exome sequencing, or now whole genome sequencing. And what's becoming clear is that we need to, again, sequence very large numbers of cases to get evidence for this hypothesis. So in epilepsy, we know of some risk alleles that contribute to a so-called oligogenic type model. These include some micro-duplications of the gene, where a small segment of the genome is actually duplicated or deleted. And it includes a very small number of genes where variation has been discovered that raises risk of the epilepsy, but doesn't in itself if you like, predeterminate, and there's a calcium channel gene that's been shown fairly convincingly to do this. But the so-called rare variant hypothesis, where a limited number of genes come together and cause the disorder, is still awaiting experimental confirmation from large exome studies that are happening now.
And with the monogenic Mendelian epilepsies, so looking at families, you can identify, and we may go into this later on, you can identify risky mutations. Is there a way to identify mutations which are protective against epilepsy?
Yeah, I mean, that's a very good question. At the moment, I'm not aware of any studies that do this. We know, however, from animal studies, particularly in rodents, that there are rodents that are quite resistant to extraneous ways of inducing epilepsy, or even indeed showing epilepsy when known mutations are introduced. So we know in rodents that there are, if you like, background genetic effects that protect you from epilepsy. I know of no experimental proof that exists in man, but it's logical to suggest that they might well do.
This might be much more important even than finding genes which cause epilepsy, because the genes, the Mendelian mutations that cause epilepsy, I mean, these are important. We can discuss this later on. But this apply only to a very limited number of cases, whereas if you could identify a protective gene, then this could show us a way to suppress seizures in general. How would one go about doing this? So in GWAS, is it possible to identify also protective genes?
Yes, theoretically I believe it should be. I don't think that's been studied, and one of the problems is that one needs, again, very large sample sizes. So, but I agree with you that might well be a very productive line of inquiry.
For the Mendelian disorders, my way of looking at them is that, I'm sure you agree, is that you can separate them into two different types. One are those that are associated with development of the brain. So brain development, if it's impaired in some way, can induce many things, mental retardation and other psychiatric or psychological effects, as well as seizures. So, and this you may be able to identify now with MRI quite easily. So this is not, I think, not so interesting to me as in my interest in epilepsy, because this is an epiphenomenon of the mild development. Whereas the other ones, which are really specific to causing epilepsy or largely causing epilepsy, many of them are related to neurotransmitter deficits or something
channels.
like that, channels. Why would these channels cause just seizures and nothing beyond? Because these channels occur quite widely in the brain.
It's a good question, and the channel genes were the first ones that were discovered that resulted in typical Mendelian epilepsies. And I agree with you that we did tend to categorise, if you like, pure epilepsy, where the phenotype was of recurrent seizures and nothing else, as opposed to the so-called symptomatic epilepsies, where there was often additional comorbidities, as you've indicated. As we've looked harder, I think we've realised that distinction is somewhat artificial. We know, for example, with more thorough studies of epilepsy in general, that there is a higher frequency of learning disabilities in children. There is a higher frequency of neuropsychiatric illnesses, both in children and in adults. And this sort of separation is really one of degree rather than being an absolute separation. And I think a particularly startling example is the sodium channel, SCN1A, which is at the moment, perhaps the most commonly identified mutation in epilepsy, where particular forms of that cause a relatively mild epilepsy with just febrile seizures, and yet other mutations in that gene can cause Dravet syndrome, a very severe epileptic encephalopathy, which is associated with recurrent seizures, progressive intellectual impairment, and generally a very low quality of life. So this distinction between what used to be called, if you like, pure epilepsy or essential epilepsy or idiopathic epilepsy, whatever word you want to use, the symptomatic is actually being blurred by more careful studies with the genetics. Having said that, there are some mutations where still the principal abnormality appears to be hyperexcitability and causing seizures, and it's present in every neuron. So, why doesn't it affect cognition? Why doesn't it affect motor control? And the answer is we don't know. An even more puzzling question, I think, within epilepsy is one might expect that a diffuse genetic disorder would cause a generalised epilepsy. And yet we know now that in fact some channel defects and other genetic disorders associated with no other comorbidities can cause focal epilepsy. And that was a big surprise, and a surprise that's not well explained yet.
And also, I think in the same category is the question of the phenotypic heterogeneity that one sees. So you've mentioned the sodium channelopathies, which cause Dravet syndrome, but in the same family, other members carrying the same mutations may have completely different phenotype. What do you think? I mean, or what do we know? How can we explain this?
So we can't explain it properly at the moment. There are two broad hypotheses. One is that it's a gene-environment interaction, and that there are some external environmental factors that perhaps the more severely affected individual is exposed to, and he or she therefore has a more severe phenotype. Despite people looking very hard for these, they're yet to be found. And that doesn't mean they don't exist, but the evidence for it is actually not very strong. The second hypothesis is that this is due to background genetic variation. In other words, in the right or wrong genetic background, one mutation may in fact cause a milder epilepsy in one individual and a much more severe one in the other. There is some experimental evidence to suggest that that is correct. That includes the fact that when one looks again at twin samples, the matching of the severity is much greater than it is in other family members, but it's by no means perfect, and there can be quite a degree of discordance in severity between affected twins. And again, in animal studies, there's good evidence that background effects have a significant effect. But what the big story is in the reason for this heterogeneity, and the main reason, we don't yet know.
And of course, we don't know what causes individual seizures to occur, mostly. So these come out of the blue at an unpredictable time in many patients, and this again an important question that genetics can perhaps not help us solve.
That's true, and we don't know, and we don't know why this is. And of course, it's one of the reasons for the misery of epilepsy. If one knew one had a seizure a month and it was on the first Monday of the month at 8 o'clock, it would be fine. You'd deal with it. But the fact that it's so unpredictable is a problem. We know, even though at the start of our discussion, we tried to make the distinction between provoked and unprovoked. We know in people with epilepsy that there are still provoking factors like stress and sleep deprivation and alcohol, etc. But most of the time there is no identifiable provoking factor, and that remains a mystery.
So you've mentioned the fact that epilepsy can occur due to genetic mutations, but also environmental factors. These environmental factors could affect the brain in different ways. And I think one of the important questions is of epigenetics. Do you deal with that? Are people studying this?
Yes, they are. I mean, my group is not specifically investigating epigenetics. I think it's a, it's a fantastic technology that is now becoming possible to do genome-wide studies on methylation, whatever, and other epigenetic changes. And there is reasons to believe that epigenetics may be one of the mechanisms by which the environment exerts its effect on the genome. But I'm not aware of any good evidence yet in epilepsy that this is the case, but it's well worth investigating.
So you're studying many different mutations, and you publish every year several new ones. Why do you collect? What's the point? Why is it important to identify still more mutations?
Before I get to that specifically, I think we've been surprised at the degree of genetic diversity that causes epilepsy. In epilepsy, I thought, and in fact I still think, we have a strength in terms of figuring out the genetics because we're able to classify the patients according to their clinical phenomenology, their EEG, etc., and we've got a fairly well-developed syndromology in epilepsy. And when this all started, we thought that for syndrome A there'd be gene A, and syndrome B there'd be gene B, etc. And of course that's turned out to be far from the truth. Syndrome A appears to have multiple genetic causes, and gene A appears to predispose to multiple phenotypes, so it has got much more complicated. So there are many reasons why it's worthwhile to continue these studies. First is in terms of fundamentally understanding the biology of epilepsy. I think it's essential to really understand all the mechanisms, and genetics is a very fundamental underlying mechanism. We hope that dilemma of the inability of the syndromes to match the genes, or the poor matching, will be resolved when we understand the pathways better. So the current major idea in the field is that although there may be multiple genes, they will all be focusing on particular pathways that will then be the substrate for therapeutic intervention. And there's already good reason to believe that might be true. For example, in the so-called mTOR pathway, which is a pathway very important for cellular growth. There are now a number of genes that feed into the mTOR pathway, tuberous sclerosis being the signal example, but now many others. And it may well be that manipulation of the mTOR pathway, which can be due, where phenotypes can be due to multiple genes, could be manipulated by a single mechanism. And we already have a drug that can do this, rapamycin, and its analogues. So I think that's a very important reason to continue the search. Secondly, if I'm a patient with epilepsy and I want specific so-called precision treatment, I want to know what my precision mutation is, and eventually that is going to be possible, we hope.
But still, so far, we've not been able to identify treatment which is specific to specific mutations, by and large.
By and large, that's correct. But there are already examples where so-called precision therapeutics is happening. One example is the rapamycin one, which I've given you. The second is that a couple of years ago, and indeed one of the Israeli families was in this paper, we identified a particular potassium channel mutation, mutations in a gene called KCNT1, which can be associated with a form of severe nocturnal frontal lobe epilepsy. And our basic science colleagues who worked on this gene and worked on the protein were aware that a drug already in use, quinidine, is actually a very effective blocker of this channel. And in the mutation, in the patients with the mutation, there is actually a gain of function. And one can show experimentally that quinidine can actually correct that gain of function. So there are now clinical trials in progress where quinidine is being used in individuals with KCNT1 mutations. Now, it's still not clear how well it works or if it works at all, but this is the way it's moving, and we're hoping that by targeting mechanisms and by targeting pathways, the genetics will really illuminate a new therapeutics. The idea from 10 or 15 years ago that we'd have gene therapy in the brain, of course, is now looking a much more distant promise. Whereas manipulating the pathways or the actual functional deficit, I think, is a realistic goal for the next few years.
And the last question, what can you say something about the contribution of Israeli neurologists and epileptologists to your studies?
Oh, it's been wonderful, and I've again many neurologists from around the country have contributed families. In my studies, I've focused on the families because that certainly has been where the money is, so to speak, in terms of finding genes in the current era. So I've had collaborations literally with dozens of neurologists from the north to the south and the east and the west to help me, and it's really been great fun, and I think very productive scientifically. And I'm particularly grateful to you for setting up the sabbatical in the first case for me in 1999 when we began.
And of course, you studied not only the Jewish population, but very much also the Palestinians.
Yeah.
You've been to Gaza at the time, and of course to the West Bank. And Zaid Afawi was a great help in that.
He was indeed, and continues to be so. So again, this was extremely important to have the personal connection with the neurologists and with the families. And I think by and large the families were grateful when we could find an answer for them. And but turning the answers into therapies is another big step, but hopefully that will come.
Thank you very much, Sam. It's good to work with you, and it's good to listen to you. Thank you very much for your work.
Thank you, Amos.